Happy Friday, everyone. Everybody wants to have extra credit today, I know. I know they do. Well, I have to be a little careful with this. There's a fairly large group of students that have gone to a meeting today who were very responsible and told me about this before the meeting. So I wouldn't feel fair giving a quiz after they had already cautioned me about this. So I'm open to creative ways of doing extra credit, though. So what are you guys thinking about that might involve them as well? Something online that a person on the video could do later. Okay. I'm open to that. Monday we should do Study Jeopardy. Actually, on Monday, I have something in mind for Monday. I'm going to do a review session on Monday, most likely immediately after this class. So then I can just post the answers, and you guys can give me the questions. Then I'll know what to do on the exam. I know what we can do. I'll tell you what. Here is what we will do. It's better than extra credit. You guys can help me to write the exam. How about that? All right? For any of you who are here, or any of you watching in TV land, if you will send me a question for the exam, I will give you credit as if you were here. How's that? That's creative. That enables everybody to do it. And I will pick a few of those questions for the exam. So that way you can help to write the exam.
Will you send us a
No. Any more than I will send you my own questions either. So, no. Okay? So, you can share them with your friends, but I would say send me your questions. Okay? Everybody get that? Everybody in TV land get that? I have to have those questions by Monday at noon. Monday at noon. Okay? Need to have your questions by Monday at noon. All right. So anybody watching in TV land, anybody in here can do that. Now, how many moms do I have here today? Oh, boy. Okay. Should we make mom do something hard, you think?
My day off.
It's your day off.
She can sing with us at the end.
So did she tell you how awful it was in here? Well, Well, I thought I prepared something that would actually help to tell you how awful it is in here, okay? So you have to sing a song, okay? So, and here we go. So this is for Mom and any other moms that are out there. It's called Oh Mommy Dear. Oh Mommy Dear, this is my biochemistry. I can't hear you guys. The problem's long, the course is really tough. That last exam, it really put the fear in me. I studied hard, I hope it was enough. But let's forget about it while you're visiting. And grab a beer when we get out of class. Then when you're gone, I'll go back to my studying. That Kevin Ahern really, truly is an awesome guy. You guys are biased. Okay. All right. So we like to sing songs in here. And we also like to talk about biochemistry, right? I know you guys like the biochemistry more than the songs, right? All right, so we've got a fair amount of ground to cover, and like I said last time, the exam is going to cover through the end of the material here. So I'm going to start talking about making proteins after we finish this, and that'll be on the third exam, not the second exam. We clear on that? Okay. So, last time when I finished talking, I gave you some background on this process called attenuation, which I tried to keep it fairly simple for you. And I want to spend a little bit of time talking about eukaryotic transcription. So eukaryotic transcription is different from prokaryotic transcription. All right? And there are actually a lot of differences. There are entire courses people take and teach on eukaryotic transcription, and we're not going to have the time to do that for obvious reasons. But I want to talk about some very general things that are different about eukaryotic transcription compared to prokaryotic transcription. Okay? So, first of all, eukaryotes have 3 RNA polymerases instead of 2 RNA polymerases. Okay? 3 RNA polymerases. So, RNA Polymerase I catalyzes the synthesis of ribosomal RNAs and some others. Okay, RNA Polymerase II in eukaryotes makes the messenger RNAs and some other minor ones as well. And RNA Polymerase III catalyzes the synthesis of small RNAs like the transfer RNAs and like some of the small ribosomal RNAs as well. Okay? So in eukaryotes, we see specialized functions for the RNA polymerases. In prokaryotes, you remember there's only 1 RNA polymerase, not 3. Okay, so prokaryotes were much simpler. All right. When we examine the promoters for eukaryotic genes, the promoters themselves are considerably more complicated. In fact, we would have trouble identifying something as a promoter like we see in prokaryotes because the sequences are very spread out, they're very diverse, and they're very specialized. Now, there's a very good reason for this, okay? Well, 3 very good reasons for that being the case. One very good reason is that our cells are differentiated. So because they're differentiated, we have cells that have different needs compared to other cells. Bone cells have different needs than skin cells, for example. And those different needs are in the form of proteins. A muscle cell, for example, needs to make myosin, which is the thing that does the contracting to make the muscles. Okay? Other cells don't have to do contraction like that. All right? So it's important that cells be making proteins where they're needed, and that means that there's a pretty fair amount of control that has to happen in terms of excuse me, helping a cell to decide whether to make one protein or not make a protein. Okay? So there are many ways in which cells, I'm going to trip on this, many ways in which cells accomplish this. Okay? And we're just going to scratch the surface of that. One of the ways in which cells accomplish that is through something called an upstream sequence element. And whenever you see the word sequence element, all it really means is a sequence. So a sequence is just a specific sequence that's found in the DNA. Okay? Everybody with me? Now, these upstream sequence elements can be located in quite a wide variety of locations compared to the actual transcriptional start. So in the prokaryotic cells, we saw transcriptional start was about 10 nucleotides downstream of the Pribno box. We also saw a minus 35 sequence, and there's actually a few others that we see as well. In the case of the eukaryotic cells, there's a lot more variability. We do see a region like the Pribno box. It's called the Tata box. Okay? And it is found anywhere from about 25 up to 100 or so bases ahead of the transcriptional start site. Okay? But in addition to that, these upstream sequences, there can be many of them, and they can be located as far away as thousands of base pairs away. Okay? These upstream sequence elements are targets for binding by specialized transcription factors. Now, you remember that a transcription factor is a protein that's recognizing and binding to DNA and affecting transcription. So eukaryotic cells have very specialized transcription factors that will be present only in some cells. So, for example, skin cells will have one set of transcription factors that might not be present in bone cells. Okay? In that way, the cell can control proteins that are needed by a skin cell, are needed by a bone cell or an intestinal cell or whatever cell it is that we're talking about. Okay? So the transcription, the specific transcriptional needs of the cell are controlled by these transcription factors that are present in that cell type. Now your next question probably is, well, how does that control happen? How do they control which of those transcription factors are made? All right? And that actually is, the answer to that question is it actually goes all the way back to development, the very beginnings of development that sets the stage for how those different transcription factors get started in their synthesis. Okay? If you think about it, a multicellular organism like us starts out as a single fertilized egg and goes from that single fertilized egg into this differentiated organism that we are. Okay? And in going through that process, we're changing from one cell type to thousands of cell types. Okay? And as those differentiated cells start distinguishing themselves from each other, that's when and how these different transcriptional factors get their start. Okay? Come on in. No? Okay. I'm not going into that class, right? It's biochemistry. What do you think we are, nuts? Okay. Now, so that's one way in which those things are controlled. All right? Now, this schematic shows a very simple, it looks very complicated, it shows a relatively simple way that transcription in eukaryotes for one set of genes actually gets initiated. Okay? So I want to just very briefly step you through it because there's some interesting things that are here. Okay? So you can see, for example, that here's this Tata box, which is like that -10 region in the prokaryotes. And what you see is that the Tata box is target for binding by one of these transcription factors. It's not a target for RNA polymerase. Remember, in E. coli cells, we had that sigma factor and we had RNA polymerase that together went and started transcription. For eukaryotic cells, it's much more complicated because in addition to needing to differentiate, I said there were three reasons why eukaryotic cells, I didn't finish that thought, 3 reasons why eukaryotic cells have the various controls that they do. One was their differentiation. Second is the fact that, remember that eukaryotic DNA is wrapped up in chromatin. It's all coiled around histones. It's a very complicated picture, and so being able to access that takes more than just an RNA polymerase to get in there and do that. So the second reason is because they are very different in terms of the DNA's access to the transcriptional machinery. Okay? And the third reason why eukaryotic transcription is more complicated is because eukaryotic DNA is enormously larger. Much, much larger. 1,000 times as large as a prokaryotic DNA. Okay? All right. So now this leads me to this figure that I'm showing you on the screen. Okay? In the figure on the screen, what you see is the initiation of transcription of one set of genes. This process starts here by binding of a transcription factor called transcription factor 2D, or it's abbreviated here TF Roman numeral 2D. TFIID. All right? That's the first thing that binds, and you'll see that it binds to the Tata box. There's actually, this transcription factor 2D is actually a multi-subunit protein itself. And one of the proteins in this multi-subunit protein is responsible for recognizing and binding to that Tata box. All right? That factor is called TBP. So TBP is a part of this TF2D complex, and TBP stands for Tata binding protein. Yeah, a lot of acronyms, I know. All right? So the TBP binds to the Tata box and holds the transcription factor 2D there. Now, everything else that's here, I'm not going to hold you responsible for. All right? But you can see that there is a sort of an orchestration that happens in terms of various other proteins that come in. And yes, they do bind in order. There's a sequence or an order with which they bind to this complex. TF2D first, then the others, then the others. And finally, after several of these have gotten there, then the RNA polymerase, in this case RNA Polymerase II, comes and binds at that place. But the RNA polymerase can't bind until this entire complex is ready for it. Okay? It can't bind until this complex is ready for it. Then, once the RNA Polymerase II gets in and does its thing, it forms that transcriptional bubble like you saw in the prokaryotes, and elongation proceeds. Okay? Okay. Yes, question? Okay, so three reasons why eukaryotic transcription was more difficult. The first was that eukaryotic cells have greater needs for different proteins because of differentiation. The second reason was because the eukaryotic cells have DNA wrapped with histones, so it's much more complex structure. And the third reason is because the DNA of eukaryotic cells is about 1,000 times larger than prokaryotic cells, at a minimum. So it's much, much larger, much more complex. Okay? Okay. The last thing I want to mention is something that's kind of cool. They're called enhancers. So I told you we had these upstream elements. And these upstream elements are targets for binding by certain transcriptional factor proteins. These proteins have a specific name. They're called enhancers. And these enhancers will do basically what you see here on the screen. Here are the enhancers that are binding to a target sequence, and that target sequence is thousands of bases ahead of where the transcription actually starts. What they do is they bind. They can actually bend the DNA around here so that they're in close connection with the place where the transcriptional start will occur. Okay? Why do they do this? There's a very good reason why they do this. It's because the DNA is wrapped with proteins. And what these enhancers are doing is helping to facilitate the unwinding of the DNA from those proteins so they can have bare DNA that the RNA polymerase can then copy. So the enhancers are binding and facilitating the unwinding of the DNA from the proteins, the histone proteins, so there's bare DNA that can be copied. Okay. And that's what I want to say there. Okay? I'm not going to talk about DNA binding proteins. I think that those are very general sorts of structural things about proteins, and they don't really tell us that much that we need to talk about here. Okay? I do want to say a little bit about the transcriptional modification of RNAs. Okay? Transcriptional or post-transcriptional modification. All right. So post-transcriptional means it happens after the RNA has been transcribed, that is, after the RNA has been made. So the things that we're getting ready to talk about are things that happen to an RNA molecule after the RNA molecule has been made. Okay? Remember that there are three major types of RNA that we talk about here. All right? The transfer RNAs are the ones that carry the amino acids to the ribosome to be translated. And they are very chemically modified. Okay? I'm going to spare you the showing of the various chemical modifications that happen. But the bases within the transfer RNA molecule have many chemical modifications that happen to them. And it's not completely understood why all of those modifications happen. We do know, however, that they are very, very frequently modified. And in a prokaryotic cell, they are the most modified of the RNAs. Okay? Now, there's a big difference between eukaryotic RNAs and prokaryotic, eukaryotic messenger RNAs and prokaryotic messenger RNAs with respect to modification. Prokaryotic messenger RNAs have very little modification that occurs to them. Very little modification to prokaryotic messenger RNAs. I'm going to show you a couple of big modifications that happen in eukaryotes. All right? So eukaryotic messenger RNAs are frequently modified. First, at the 5' end of eukaryotic messenger RNAs, there is a structure called a cap that is put onto it. A cap. And what the cap is a modified nucleotide. It's actually a guanine nucleotide that is linked to the 5' end of that messenger RNA, okay, in a very unusual 5' to 5' linkage, meaning the 5' end of the modified base is linked to the 5' end of the messenger RNA. This has the result of creating a structure that looks unusual to the cell, and it turns out to serve as sort of a flag for the cell when it comes to the making of protein. And we'll talk about that later. But suffice it to say that the 5' end of eukaryotic messenger RNAs are very different than prokaryotic messenger RNAs. Prokaryotic messenger RNAs are not modified at the 5' end. Eukaryotic messenger RNAs are. Yes, ma'am?
The modified one
Yep.
is attached to a regular mRNA.
So this is the regular mRNA that's running down from this point. So this is the 5', this was the original 5' end of the messenger RNA, and this 5' end has been attached to this unusual nucleotide up here. So, no, it's not another, it's just a modified nucleotide that's put on the end of it. Yeah. Okay? Okay. Question? Clear as mud? Want me to say it again? Yeah. Okay, I'll slow down. All right. So, what we're talking about here are messenger RNAs, right? So messenger RNAs are the things that carry the message to the ribosome that gets translated, right? This is one of the 3 types of RNA, right? And this is the one in eukaryotic cells that's made by RNA Polymerase II. Okay? Now, what I'm telling you is that when the RNA polymerase comes along and it decides, okay, I'm going to start copying and making this messenger RNA, what's it going to do? Well, base number 1, nucleotide number 1, is right there. And it's going to synthesize a whole string of these nucleotides going all the way around the room. Okay? Now, so there's base number 1, base number 2, base number 3, 4, 5, 6, 7, 8, 9, 10, et cetera, et cetera. Okay? Now, after that first base is made, there are enzymes in the cell that will come along and modify that first base by adding an unusual nucleotide to it. Okay? No? Okay.
So that's just, I'm just not
This
understanding.
right here?
Yeah.
Okay. So let's say so you understand that the RNA polymerase started making this, right? Okay. So if I had a prokaryotic cell, that would be nucleotide number 1, and it would just go along like that. And that would be all there would be, right? But in the eukaryotic cell, after this thing has been made, some enzyme comes along and grabs this thing and says, I'm putting this cap on the top of it, just like I put a cap on my head. So it's just linking this guy to this.
So it's like a random, it's just a single base.
A single base, that's right.
So then now this has 2 3' ends?
So this is a good question, actually. This guy has essentially 2 3' ends because it has a 3' in there because this is a 5' to 5' linkage, and at the other end there's a 3' end. You're right.
And so does this always happen or just like,
It always happens in eukaryotic cells.
so after it's made,
That's right. So the first step is making this. You're right. The second step is putting the cap on it. And the third step will actually be, it will be translated into protein. Now, this putting the cap on turns out to be essential in eukaryotic cells for it to be translated to protein. Okay. Sorry if I'm not being clear. Other questions? Don't be afraid to ask questions. Yeah. It's always the same nucleotide. So this unusual, this is actually a modified form of guanine. This unusual nucleotide is always the thing that's used to put the cap on. Right. Other questions? Yes. Why is it important? It'll become more important when we talk about translation. But for our purposes right now, it's just a way for the cell to identify what a messenger RNA is. This only happens to messenger RNAs. It is a distinguishing feature. That's right. Yeah? So what it's doing is this is literally a flag. It looks unusual to the cell, and this provides a handle for proteins in the ribosome to grab a hold of. Nope. Messenger RNA versus a ribosomal RNA versus a transfer RNA. Okay? Okay, good questions. You guys are always so quiet. I'm not used to this. This is good. But Friday must be, maybe the sunshine is bringing it out on you, right? Or Mom's weekend, maybe? Mom, maybe you're inspiring everybody today. Have you thought about it? Yes?
So the guanine nucleotide that's linked up top, does the fact that it's guanine, does that actually have anything to do with what we're talking about, the 8-oxyguanine?
No. Okay, so her question is the fact that this is a guanine related to that oxidative form of guanine I talked about the other day? No. So it's just a guanine. Yeah. But it's a modified guanine, but it's not the same as the 8-oxyguanine. Okay. There's more. There's always more, right? So the 5' end of these messenger RNAs is getting modified, and the 3' end gets modified too. The 3' end is probably easier to understand. Let's take a look at what happens at the 3' end. Oh, actually, it's not even on there. All right. I'll have to tell you what's at the 3' end. 5' end, we got a cap. 3' end is hanging out. What's that? Well, the other end, the other 3' end, not the guanine end, the other end of that messenger RNA, there's something called a poly-A tail that's put on. Look at this. Good. I should have given you a pop quiz on this. All right? Meaning that at the other end of the messenger RNA, there's a string of about 200 to 300 adenine nucleotides that are added to that end. 200 to 300. Anybody want to guess why? No? Okay. Well, I'll tell you. You're probably not going to guess it anyway. All right? It turns out that it's to help stabilize the messenger RNA. Okay? To stabilize. What's that? Who guessed it? Did you guess it? Okay, I'll give you 1 point of extra credit for the exam. Give me your name and I'll make sure you get it, okay? It's stabilizing it. Oh. It's called the poly-A tail. Poly-A. Okay? Now, I'm not going to go through how it stabilizes it today, but I will talk about that later, all right? Suffice it to say that there are enzymes that break down RNAs, and this helps protect against those enzymes. Okay. The last thing I want to talk about that happens to mostly messenger RNAs, but it also happens to some of the other RNAs, is a phenomenon called splicing. splicing is a process that happens, again, only in eukaryotic cells. It doesn't happen in prokaryotic cells. Okay? Splicing, of course, I'm so old, I remember when actually tape recorders had tape, right? That's where they got their name from, right? And in the old days, if you wanted to put together two pieces of sound, you actually had to take a razor blade and cut the tape, and then go over here and cut it another place, and then put the two pieces that were left together with a piece of tape. And so that way you could put one sound segment next to another sound segment. And that phenomenon was called splicing. Well, the cell is doing splicing in the sense that it's taking a messenger RNA, it's cutting some of the inner pieces out and joining the other pieces together. So the inner pieces that get removed have a name. They're called introns. And the pieces that get joined together have a name, and they're called exons. So introns are the parts of RNA that get spliced out, and exons are the parts of RNA that remain. Now, if we're talking about a messenger RNA, you can actually see the finished version of the messenger RNA at the bottom. There's the cap at one end. There's the poly A at the other end. And there are the exons in the middle that have been joined together. Okay? I think we've reached the end of the material for the exam. Finally. What's that? Do we get out of You don't want to sit and listen to me for the next hour? I was planning to lecture until 5:30 today. Unfortunately, we can't. Well, we could if we went really fast on the next time. No? You don't want that? Yeah. Okay. All right. So, end of material for exam 2 right there. All right? Let's talk. I'll go reasonably quickly, so we'll get out reasonably early. How about that? Okay? All right, so we've talked about 2 of the 3 processes of the central dogma. The first process, of course, was the DNA replication. The second process was transcription. And now we're getting ready to talk about translation. And translation, of course, is the synthesis of protein. All right. The process of synthesizing protein is a very intricate one. But like transcription, we see it occurring in about 3 phases. There's one other phase that's added here. But you can see initiation, elongation, and termination. Just like you saw in transcription, only now we're making protein instead of making a messenger RNA. Okay? The other thing that has to happen before initiation is that amino acids have to be what we call activated. This is actually fairly simple. Amino acids have to get attached to the proper tRNA. They have to get attached to the proper tRNA. What's the proper tRNA? Well, everybody's heard of the genetic code, right? The genetic code is a 3-base sequence that corresponds to each of the 20 different amino acids. There are 64 codons. There are 20 amino acids. That means that most amino acids have more than one codon that can specify them. So a codon is a 3-base sequence that specifies an amino acid. Now, students get confused. Where's the codon? Is the codon in the messenger RNA, or is the codon in the transfer RNA? The codon is in the messenger RNA. There's a complementary sequence to the codon in the transfer RNA. So the transfer RNA has a complementary sequence to the codon, and it's called the anti-codon. Codon, anti-codon. Okay? What's that? Where's the codon? The codon is in the messenger RNA. Okay? All right. So we're seeing the genetic code here. If we look at the genetic code, I think you should memorize this for the exam, right? You guys are getting good. I can't play anything over on you. All right. There's a few sequences I think you should know. All right? AUG. AUG is known as the start codon because all proteins start with that sequence, AUG. And if you look on here, you see A, and you see U, and you go down to G, and you see, oh, it's methionine. And that tells you that methionine is the first amino acid of all proteins as they're made. Methionine. AUG. So you should know what AUG does. It's called a start codon. There are also 3 codons that are known as stop codons. And what they tell the cell is we are at the end of the coding, stop making protein. Okay? There are 3 of them, UAA, UAG, and UGA. And those are the only codons, those 4 codons, the only ones I think you should know. UAA, UAG, and UGA. Now, I mentioned that you can look at this and you can see, well, here's valine. Valine can be specified by GUU, GUC, GUA, GUG, meaning that valine could be specified by GUX, where X is any nucleotide, right? The code is what we call redundant, meaning more than one codon for many amino acids. The code is organized in an interesting fashion. Okay? Look at up here. UUU, UUC specify phenylalanine, UUA, UUG specify leucine. Or let's look over here at, let's say, GAU and GAC specify aspartic acid, whereas GAA and GAG specify glutamic acid. Okay? Aspartic acid is specified by GA pyrimidine. Glutamic acid is specified by GA purine. There's balance and organization to that. The third base of the codon is what people talk about the wobble position. Because the third position isn't as important as the other 2 in determining which amino acid gets put in. If I have GU anything, I put valine in. Look at aspartic acid and glutamic acid. They both have R groups that have carboxyls. GA blank means I get an R group carboxyl. There's neat organization. Now, you're not going to memorize these. I'm not asking you to memorize them. Okay, that's not the point. But the point is to show you that with this redundancy, that mutations that occur in the third base don't have as much importance as those that happen in the first 2 bases. Okay? And that's sort of summarized down here at the bottom. All right. Now, I said I would go fast. I said I would try to get you out of here. So what I'm going to do is I'm going to talk about amino acid activation, and then I'm going to just very briefly start you with the process of translation, and then we're going to finish. Is that okay? Okay. So, amino acid activation. Let's talk about that. Amino acid activation is the attachment of the amino acid to the proper tRNA. What's the proper tRNA? Well, that depends on the sequence of the anti-codon in the transfer RNA. To refresh your memory about the anti-codon is found, oh, that's the wrong structure. The anti-codon is found. Oh, I don't have it on here. Okay. The cloverleaf structure I showed you, I thought I had it on here. The cloverleaf structure of the transfer RNA has a loop at the bottom. The loop at the bottom is called the anti-codon loop, and that's where this 3-base sequence complementary to the codon is found. That 3-base sequence determines what amino acid is going to be attached at the other end. The attachment at the other end is the activation step I told you about. There's an enzyme that catalyzes the attachment of that amino acid to that transfer RNA. It's called the amino acyl-tRNA synthetase. You want to write that down real quickly? No, you probably don't. I'll let you name it. You tell me what to call it. What should we call it? Something simple that you will remember. I'm serious. So the enzyme that attaches the amino acid to the tRNA has a long name called the amino acyl-tRNA synthetase. And if you want to remember that name, you can. Or if you want to call it Larry or whatever, you can. But we have to decide on what it's going to be. So that's what we're deciding right here. You want to call it Kevin? Okay. One vote for Kevin. I wouldn't propose Kevin myself, but okay. You want to call it Kevin? Okay, so we'll call that enzyme Kevin. If you call that enzyme Kevin on the exam, you will get counted correct. I'll have to make sure the TAs understand when everybody talks about Kevin what the hell they're talking about. But okay. So Kevin attaches that sounds kind of funny, attaches the amino acid to the tRNA and attaches it to the proper one. It turns out that there's one Kevin for every, you guys started this, one Kevin for every amino acid. So there's a Kevin for alanine, there's a Kevin for leucine, there's a Kevin for lysine, etc. That means there are 20 Kevins, and that's a scary thought in itself. Okay? All right. The process of translation occurs in a specialized structure of the cell called what's the structure that makes proteins in a cell? Ribosome. Okay? The ribosome is where translation occurs. Okay? Ribosome has about 50 proteins in it. It's got 3 or 4 ribosomal RNAs, depending upon whether it's prokaryotic or eukaryotic. And as I mentioned before, one of those RNAs catalyzes the formation of peptide bonds. Now, I promised to finish early, so that's all I'm going to say at that point. I will let you go, but one qualification. Yes? Kevin attaches an amino acid to the proper tRNA. I have a song to sing this, and you have to sing loud. And if you sing loud, there will be an extra credit on the final exam, on the next exam, whatever the exam is. All right? In fact, this is an important song. This is actually the national anthem of biochemistry. So I want everybody to stand and put their hand on their heart. Put your hand on your heart, please. This is serious. Come on, guys. All right? It's to the tune of America the Beautiful. Oh, beautiful with RNA to make the peptide bonds. You hold tRNA so it can pair up with codons. The ribosome translate mRNA, initiate and translocate from start to UGA. Have a fun weekend.